So small molecules could be sugars, could be fatty acids, could be amino acids, could be a lipid precursor like cholesterol, etc. As long as the molecular mass is below 1500 units, we can classify them as small molecules. Again, I'm trying to avoid as much chemistry as can be. Okay. Now, we agreed that living cell is a factor. Any question? Okay. So the living cell is a big factory, really. Okay. And a massive network of interconnected systems. Okay. Pretty much think of it as a very busy city, like London, for example. Okay. Too many busy roads, too many vehicles, kind of circling around the city, etc. And it all needs organization, traffic control, otherwise we will end up in a mess. Okay? And there is no time you had signs that the traffic is going crazy within your living cell. Everything is just running as normal. Otherwise, you will feel ill if the traffic is not going well. But the issue is, as you can see, is a very complex network of systems, making their study not really a straightforward process. Okay? Too many proteins involved, and the proteins are the catalysts. Okay? What really speeds up the reactions. And too many small molecules produced or consumed. Again, this is making the whole process very complex. And we pretty much need a way to study such a complex system. So how can we study such a complex system? And again, the whole concept of studying such a system is not really new. In a way, it's as old as our species, okay? For example, more than 2,000 years BC, Chinese medicine used metabolomics in a way as, okay, they can look at the smell of the urine, etc., to test or to tell if there is a disease state or not. So, in a way, this is metabolomics, okay? However, modern metabolomics, as we know it, maybe is about 50-ish years old. So, the author of this paper, Linus Pauling, this is a really big name in chemistry, okay? This guy, kind of one of the founding fathers of modern chemistry as we know it, okay? And he was interested in a very diverse set of topics within chemistry. Unbelievable, really. But again, I was kind of intrigued to see him also interested in metabolomics. So as early as 1971, he used gas chromatography, okay, to look into maybe, again, something similar to the Chinese medicine as the characteristics of urine and how it can actually tell about a normal or disease state. So the take-home message is metabolomics is nothing new, is something very sensible to consider, and has always been of interest to the chemical biology community. Okay. So, obviously now, if we look into the set of small molecules within the cell, in whatever form, either in biology of disease.
Is it the introduction?
Yeah.
Oh, okay.
No.
Which one sounded like just like?
Go to canvas.
Yeah, I did, but I think it's not being updated.
Oh no, don't worry, this is very something. So the lecture material is very new, it's not on canvas yet.
Yeah, that's one.
Yeah, good morning everyone. So I didn't really upload the content, but you will have it shortly after the lecture. Okay. So again, you can think of an illness or malfunctioning of the cell as a way to perturb the system. Okay, something changing the normal behavior of the cell, and this kind of change is reflected in the content of small molecules. So by studying the set of small molecules available and their concentrations, etc., you can use this to tell if there is a specific disease state or not. And obviously, this change is not necessarily due to a disease. You may be taking some drugs, and taking the drug, for example, you will find once you take the drug, the coloration of your urine is different, okay? Or the smell is different, or you smell differently, etc. Okay? It happens in many cases. You may find yourself, for example, once you take a drug, you sweat more. You kind of, your saliva is running maybe higher or lower as you feel kind of dry mouth, etc. So all these changes are actually perturbation to your system, and metabolomics can help to kind of identify such changes. So metabolomics is very widely used nowadays, not necessarily just in kind of detection or identification of a disease state. It can be applied in so many areas. You guys are good here?
Yeah, sorry, I'm just confirming something. I just didn't properly.
No worries. If you have a question, just ask. So as you can see, okay, metabolomics really can help with so many fields and techniques, etc. Okay, and maybe again, my advice always for you guys as always get outside your comfort zone. If you are being trained as a biologist or a biomedical scientist, know something about, for example, related fields like chemistry, etc. Because what I noted is those who are studying chemistry have little knowledge / appreciation of biology, which is obviously so wrong. And vice versa with regard to whoever is studying biomedical sciences or biology. Simply because most of the disciplines we are studying now are of interdisciplinary nature. And most of the opportunities available in the market are also of interdisciplinary nature. So you need to get out of your comfort zone and know, for example, okay, a field like metabolomics, how it operates, how I can contribute to. Again, not necessarily you are getting totally away from your area of interest or expertise. You are building on the same area. So metabolomics can be applied in so many fields nowadays, okay? And what is really of interest to you guys is, can we use metabolomics to identify specific biomarkers for disease? A biomarker is a small molecule which you can monitor, and it can tell you, okay, that a specific disease state is there or not, okay, as we will see later. For example, if you look at the cholesterol level within the cell, can it tell you about a specific disease state or not? Okay, if you are maybe, we are kind of, everyone now is interested to be in a healthy shape, maybe taking some nutraceuticals, for example, okay, amino acids, etc., for a muscle building, etc., or just caring about your shape, etc., okay? But obviously, you don't know if whatever you are taking is good for you or not, okay? And metabolomics can help here as, okay, you can take whatever certain of mixture of amino acids, for example, and then you can look if this is really helping you or not. Maybe you can identify that whatever is missing within your system is maybe a aliphatic amino acids, okay, and you are interested in building some muscles, so you should focus more on aliphatic amino acids, or maybe whatever your kind of everyday food is missing is aromatic amino acids, etc. Or no, you actually need a mixture of arginine and alpha ketoglutarate, etc. So you can use metabolomics to guide what sort of nutrition or additives you can add to your food or kind of training exercise routine to get to your destination, as if you are looking at a specific, for example, body, or maybe treating a specific disease, etc. And most importantly, now, personalized medicine, okay? So this is pretty much where our civilization is heading in terms of the next target in making things as super customized. As pretty much in the near future, everyone will have their DNA sequenced, okay? And then you can pretty much work out 10 years in advance that you have potential to develop diabetes, for example, or a specific disease state. Touch wood, cancer will never be the case, etc. But maybe if you have certain genes, then the DNA sequencing can tell you that, okay, there is a possibility. So obviously, knowing the role of these genes and what they can play, etc., you can counteract their effect, for example, by providing a small molecule or trying to make some modifications, okay, to alleviate or eliminate this possibility. So again, metabolomics can really be helpful with regard to personalized medicine. Now, obviously, the question that comes to your mind is how a metabolomic workflow is looking like. Again, I'm trying to take as much complexity and removing all the chemistry jargon, etc., out of this. But the concept is very simple. Pretty much as you go to your GP and they ask you for a sample of your blood, urine, etc. So you start with a sample, and the sample could be some body fluid or some body tissue, etc. And always the question would be how to extract the stuff of interest out of it. And this is where you need some appreciation of chemistry, because most of the time you end up using specific adsorbents, something that can take away the small molecules and remove the proteins, the DNA, RNA, etc. So, going back to the first slide, you had genomics, transcriptomics, proteomics or proteins, and you have small molecules. This is pretty much the mix that is present within any body fluid or within any living cell. But out of this mixture, you don't really need anything but the small molecules. So most of the time, you have your sample, and you have to extract your sample, okay? Once you extract it, and is in a form which is near ready for an analysis, you may have to treat it in a way or another using chromatographic techniques, which again I'm not going into their details, but this is something for you to know, that there could be some pre-treatment before submitting your sample for analysis. Once your sample is ready for analysis, then you can use whatever analytical technique in your arsenal. It could be mass spectrometry, it could be liquid chromatography, mass spectrometry, gas chromatography, mass spectrometry, it could be nuclear magnetic resonance, NMR, etc. Obviously, coming from a biomedical background, most likely you will be in a lab. Patients will be submitting, or GPs will be submitting samples of patients to you, and you need to know how to look after the samples, okay? And then how to do the pre-treatment to get the samples ready for analysis. It depends where you are based, you could be actually asked to do the next step, i.e. the analysis itself, using mass spectrometry or NMR, etc. And obviously you will be trained on using these techniques. But having knowledge and appreciation of what they are about, etc., can really help. I'm not going to explain the concept of mass spectrometry or NMR, etc. I will leave it for you guys. But these are very fine analytical techniques, okay? And the beauty of them is they can really deal with a very complex mixture of small molecules, especially NMR and mass spec. It's pretty much like taking a big photo for the entire team here, and with the capacity to go from the big picture into a focused one and tell who this person is or who that person is. Make sense? And obviously the interaction and the concentration range, etc. The issue always is, once you finish the analysis, you will end up with a forest of data, okay? And you need to know how to deal with such kind of forest. How can you decomplex and make use of it? And this is where the statistical analysis usually takes place. In a way, eventually you end up producing something like a heat map. And this heat map will tell you that, for example, anything that is red is out of range, higher than, green in range, yellow below range, etc. And this could give an idea as, okay, you have 1, 2, 3 metabolites which are higher than the expected range, 10 which are in range, and 5 which are lower than the range. Yeah.
I was wondering if you had any recommended readings for
Yes.
this module, because all I could find was one book on critical thinking skills. It sounds like this topic's going to be very topic heavy, so I was just wondering if you could give me some.
Don't worry at all. You will have the slides, and within the slides there is recommended reading. I will mention. Apologies if I didn't refer to them earlier. Here is maybe one nice tutorial you can start from, and there you will find more reading material if you wish. Again, the target or what I'm going to examine you on is not really the heavy kind of details about analytical techniques needed, etc. It's just more of the appreciation of the concept itself, okay? So that in a way, if you ever be in an environment where you have to deal with metabolomics, is not something totally new to you. And obviously, most of the modern kind of tools for detection and quantification of any form of disease rely on metabolomics. Okay, so don't maybe take it as a daunting task. Just maybe wait until we finish and look at the available reading material, ask questions, etc. And I'm sure you will find it much easier than you would expect. Okay. So eventually, whatever heat map you will be producing will help you work out, okay, what is going on. This is, for example, cancer. What type of cancer? Okay, and could help with the treatment protocol, etc. Now, as we agreed, we have 2,500 small molecules. So, whenever we look at them, are we going to take a global approach, i.e., we will look at the 2,500 metabolites, or we actually need to look at just specific kind of metabolites? The two approaches actually exist. So you could take a global or untargeted approach, It depends on the condition you are looking at. Or you can just take a targeted approach, okay? As you are interested in looking at a urine sample and looking at the metabolites within the urine sample. So you can either take a global or untargeted approach, which is obviously more complicated, or you can take a targeted one. So, for example, as early as 2002, working out a complex disease state like coronary heart disease was actually enabled by a nuclear magnetic resonance, which is a very kind of famous technique in a chemistry based on the magnetic properties of the small molecules within your body. Okay. Again, it's a very complex disease state, and you can reach some quantification and characterization of the state using maybe a technique which is not very difficult, but equally not very easy to deal, except if you have the right maybe background with it. Here is another example for a application of metabolomics in cancer, as they were looking at a number of small molecules. Here you can get a list of them, mostly amino acids, etc. And the heat map produced has been produced actually very intriguingly, just within 35 minutes. So you have the sample, whatever the sample is, you do the microarray analysis, and you get the result within 35 minutes, which is amazing, okay? And it will provide you with a heat map with a specific set of small molecules, and it will tell you if you have them within normal, below, or above normal concentration, or if they are totally absent, and then you can work out for patient one, for example, if a specific type of cancer is there or not, etc. So just maybe as a way for you to appreciate how useful metabolomics could be. Again, the cancer cell has a very unique metabolic network, and it really differs from the normal cell with regard to how much ATP it is consuming actually, and what type of amino acids are being kind of consumed or in heavy use by the cancer cell, okay? And in a way, this is very intriguing because again, the amino acid transporters within the cancer cell are looking for specific type of amino acids, for example, like glutamine, etc. So potentially, one way to treat such types of cancer is to provide the cell with a molecule like glutamine, which the cell will be tricked to take in, try to use, but rather than being able to take it and make a protein, it will pretty much stop the activity of the cell, of the cancer cell. And this is something, for example, my group is looking into as, okay, we produce some small molecules similar to glutamine, which can trick the cancer cell if they are kind of engulfed by cancer cells and stop their kind of metabolic activity. And because the cancer cell is very active and takes such type of compounds heavily, unlike the normal cell, then the process can be selective. And this is a major issue when it comes to treatment of cancer, as how can you differentiate between a normal cell and a cancer cell? Most of the time, whatever treatment is used actually kills both, as normal and cancer cells. So, by understanding the metabolomic network within a cancer cell, what type of small molecules it is using the most, etc. One, you can identify, quantify, what cancer is there and what stage it is at, and also it can help with recommendation of treatment, etc. And this is a paper actually about use of carbon-13 flux analysis to help cancer biologists sort of characterize what sort of cancer is there in a specific patient and potentially what stage the cancer is at, etc. So again, all in all, metabolomics is in heavy use when it comes to characterization or differentiation between a normal state and a disease state. Okay. One more example, as you may, for example, be sitting in a lab and you have a sample of serum cholesterol, and within the sample you find high level of cholesterol. But again, not necessarily having high levels of cholesterol is indicative of a specific disease state, simply because high levels of cholesterol can be really attributed to a number of mechanisms. Okay, the first one, it could be just whoever the patient is, pretty much their absorption of cholesterol is really good. So you will find high levels of cholesterol within their system, but not necessarily this is related to a specific disease state. Okay. Also, the person could be producing too much sterol or cholesterol within their system, okay? Simply because their liver and associated organs, etc., involved in cholesterol formation are very active. Thirdly, it may be just the person is not really using exogenous cholesterol and being able to convert it into bile acids, etc. Okay? So, having high levels of cholesterol could be due to a number of mechanisms. So, metabolomics can help you work out which case out of the 3 case scenarios presented to you the situation is, and also can help you provide the right treatment. So, for example, if the person is really having high absorbance levels of cholesterol, okay, then potentially you can interfere with the mechanism of cholesterol absorption, okay, i.e. you can provide them with something that can work within the intestine and decrease the absorption of cholesterol into the systematic circulation of such individuals. Make sense? However, if the person is really, their kind of system is producing too much cholesterol, then you can provide them with inhibitors of cholesterol biosynthesis. Obviously, providing inhibitors of cholesterol biosynthesis to the first group wouldn't help their case, okay? And vice versa, okay? So to know the right treatment, you really need to know which mechanism is not operating or operating in a specific way so that you can engineer the treatment. So again, these are just examples of the utility of metabolomics in a biology of disease, etc. So what is expected from you guys here is to know the terminology, okay? The various terminologies we have been through. For example, what a small molecule is, what is metabolomics, what it can be used for, what are the various stages of a metabolomic workflow, etc. Okay, certain applications, etc. The exam questions will be very much related to the content provided to you. Obviously, we will meet again during the workshop, etc., and we will study at least 1 or 2 papers. Look into them, have more examples, more specific examples, etc. And then also answer any questions you may have. So, any question for today?
Sorry, I might have missed it at the start, but how are the exams? What is the exam like? How are we assessed basically?
So, generally speaking, for the overall module, the questions are very much like MCQ type of questions, okay? And generally speaking, is all about interpretation of data, okay? So you have, for example, maybe like the cancer kind of heat map shown to you. Maybe you can have something similar, and you can be asked as which disease state you are, you think the case is. Obviously, today is just an introduction. We still have more sessions to go through, etc.
Is it open book or closed book? Do you know?
Closed book.
So is it on campus?
Yeah. Yep.
You were going in detail about metabolomics. Will that be something that's carried throughout the module, or is this?
No. This is just specific to my part.
Okay.
Other lecturers, very much their material is already on Canvas. Okay, so you can look at. At least I will see you another time, as next week we have a lecture and then a tutorial, etc. So this will be related to metabolomics. But for example, other colleagues will be teaching you the topics that you can find on Canvas. Yeah, it will be done as soon as today.
With the poster presentation, is that done in front of the whole class, or is that done at set times?
Yeah. I will get back to you on this one. Any more questions, guys? Again, we will meet again next week and we can discuss more. Okay? If no more questions, see you next week then.
Everyone I spoke to about biology disease, people that are done biology. Everyone I spoke to about biology disease in the previous 2 years, they told me